NADPH PDF.
Meeting is being recorded.
Okay. Okay, you can record the meeting if you want. So I just open my that one result protein PDBQT. Okay, just opening it. Where is the 7 km PDBQT? Okay, gone. So many files. Yeah, this one Autodoc structure. Okay, yeah. So your structure is renamed as Autodoc structure. Okay. So this one is your PDBQT file. You have renamed this one, na? You have renamed this one, na? 7VM with NADPH.
Yeah.
Okay, so this is the PDBQT file. Now I will open the out file. PDBQT. Why there is no need of cmd.psc. I want PDBQT out file. Okay, this one is PDBQT file. Just download it. Okay, now I'm just opening PDBQT file out file. Okay.
Okay.
Now you can see this one.
Yeah.
Okay. Now what you do? You are taking as a fifth number conformer. Okay, you just do it for all the conformer. Okay, and share me the, and then you look at the interaction. Okay,
Yeah.
but for now I will so because see the first one, second one is the same, third one is little bit opposite orientation. In the paper they have mentioned that this one is in opposite orientation, na?
Yeah.
So this one is the right conformation. It may be oxygen in this side, and here oxygen in the that side. Okay. So this one is the third one. This is the same fourth. This is the fifth one. Sixth. Which one has the high energy? 7 is not, 8 is wrong. Ninth having.
High energy.
This is one is also wrong. Which one has the high energy? Fifth one, sixth. Wait a minute.
I have to see the log file.
So first having high energy, I think, है ना?
Yeah.
First having high energy.
17.9. Yeah, first one.
Second, how much the difference of energy? Second one?
0.317.
0.137. This is the energy.
Minus 7.8.
What you are saying 0.1?
Affinity. Ki,
Energy I am talking about, na kilojoule per mole.
yeah.
7.
Minus 7.8.
Yeah. So this one, third one is,
Minus 7.7.
fourth one.
Minus 7.5.
So all are same. Okay.
Mm.
So I think this one is not good. I think this one is fine. This one is fine, no?
Mm.
Second one having high energy.
First
I will
one
take,
is. First
I will
one.
take the first. I will take the first one. Okay. Okay.
Mm.
And you have taken the fifth one.
Yeah.
You have taken the
Fifth one.
you have taken the fifth one. I will try. I will take the first one. Okay. ठीक है कोई दिक्कत
नहीं.
कोई प्रॉब्लम नहीं ना नाउ गो टू फाइल एक्सपोर्ट मॉलिक्यूल ठीक है सेव द फाइल एज पीडीबी सो आई सेव द फाइल एज फिफ्थ नंबर पीक्यू 7 VM ठीक है 7 VM आई सेव दिस एज ए नेम हो ना आई सेव इन द फोल्डर डेस्कटॉप okay I just save it in the desktop now I'm just reinitialize the session. Okay, now I'll take the that dog complex 7 conformation. Fifth conformation. I've just opened it. You can see this one.
Yeah.
Okay, this one I have taken. Okay, now so just change the color if you want. Go to display background white, change into blue or something else. You can even preset. There is option called ball and stick. You can change it to ball and stick. Color it with yellow or mark by item. Okay, you understood this one? So this look good. Okay,
Yeah.
change the color of the chain if you want. Yeah, okay. Okay, this one. Okay. Now your protein NADPH in ball and stick. Change it into ball and stick if you want. Preset ball and stick. Make it same color, blue, and change by item. Color by element. You understood this thing?
Yeah.
Okay. Now open your which one? One keyword structure. So I've opened the one keyword. Okay.
Yeah.
Now what you do? Go to action, delete the water, remove water molecule. I've removed the water molecule. I'll go to select chain, go to display sequence mode. Now I will remove the B chain of this one. First you select this one. This is the B chain. And then don't remove sulfate ion because this one important. Don't remove NDP from A chain. Remove this to B chain. Just remove it. Now you are left with 7 VM docked penetrant quinone, one keyword with NADPH and sulfate ion. This is an experimental crystallized structure. You have here experimental NADPH structure. है ना? Now what you do? Just select this one and change the sewage stick. Okay. This one is the sulfate ion. That one is bind very much close to the nicotinamide. So we can say that this sulfate ion, where what are the residues that are interacting with this sulfate, acts as a putative substrate binding site. You understood this thing? Because this protein is not crystallized with the quinone. But we know that the substrate bind very close to the nicotinamide ring. So we can say whatever the residue that are interacting with this sulfate ion act as a putative substrate binding site. Putative, why putative? Because we don't know. That's why we say the term putative. You understood? Hello?
Hello. Yeah.
So
Got it.
say na yes or no. Okay.
I was saying, but I was muted.
So why you muted it? Don't mute it. Now change the color. How do I know you are understanding or not? Okay. You have to say
Okay.
yes or no everything. Okay. Sulfate is already SO4. Now what you do? You superimpose this one to this one or that one to that one. Anyone you can do it because that time you have not saved this protein as a. That's why it's moving from here to here. Now you can superimpose both one. Go to action. Align to which one you want to superimpose. This one to this one. So we have a experimental structure. Okay.
Mm.
We know this is experimental. This is hypothetical because we have dogged this one. But this one we know that this one is experimental and we have the substrate. So we have a reference molecule substrate. Then what we will do? We will dock this molecule to that one. You understood why I am docking superimposing this one to that one because we have a putative substrate binding site with reference to sulfate ion. That one is experimental crystallized structure with a sulfate ion. You understood this thing, na?
Yeah.
So we will dock that one to this one. Okay. So
Mm.
this one will take as a reference molecule. So you align 7 VM to
1
1
cube.
kilo. So I will align both one.
Mm.
Now you can see here the alignment.
Yeah.
You can
Little
see the alignment. Okay, wait
difference.
a minute. Wait a minute. Now what you do? Now tell me which residue are interacting with sulfate ion. In case of 1K, or do the protein plus.
Protein
Okay.
plus is not opening in my.
Okay, no problem. I can do it here. I just minimize the one structure first and then mark all the residue. Okay. Then hide all the cartoon. So
Yeah.
I just first hide this one. I will work for this one. Okay.
Mm.
Go to this one. Select this one. Okay. I've selected this one.
Mm.
Or you can do what you do. Go to this one, 7, this one QR. Go to Excel, reset technical. Okay. Now you can see I can mark the sulfate ion interaction. What residue are interacting with sulfate ion? Because this one is a putative substrate binding site. What are the residues that are interacting with the sulfate ion? Act as a putative substrate binding site. Okay.
Yeah.
We can take the difference from 7B, PQ, the octet structure. Now you can compare whether what what are the residues that are interacting, whether they are really a substrate binding site or not.
Mm.
Okay.
Mm.
So we can superimpose, then you can understood. Now you can see this one is interacting. Sulfate is interacting with these two. Sulfate is interacting. We don't want. We want the residue only. Okay. So you can see this sulfate is interacting with this residue, two residue. One residue is this one. So you can zoom it if you want. Now you can see this sulfate to oxygen is interacting with this residue. Okay, if you change the residue, this one. Okay, and then again, which residue are interacting? This one, this one. Okay,
Yeah.
or so these two, or no one is interacting. So this oxygen is interacting, this we don't want. This oxygen is interacting, this one we don't want. Okay, there is no interaction with this oxygen.
Mm.
Okay, so this oxygen is interacting, we have already marked, and this oxygen is interacting with this residue, already marked it. Now
Mm.
what you do? And see, you can, this oxygen is also interacting with this one. We don't want. Okay. Now you just change into stick. So it's stick. Got it?
Yeah.
Label it. Label the residues. Okay.
Mm.
So you can see the conservation. See, tyrosine 52 come here. It means the tyrosine 52 is conserved. So what we are protecting the correct.
Mm.
Go to scene. Okay, go to setting, label, size 18, go to setting, label. So it means the tyrosine 54 is a very important residue. You can, if you mutate tyrosine 54, your activity will go. You understood or
Yeah.
not?
Yeah.
So now I can see it, I can show you the tyrosine 54 from other one also.
Mm.
Now what you do?
Remove
So
it.
we already got this thing.
Mm.
Okay.
Mm.
Now it's just convert into cartoon. 7B is this one. Oh, sorry. This one is don't want. Where is 1 QR? This one is 1 QR.
Mm.
So it's cartoon or hide cartoon. You can do it. Okay.
Mm.
Or you can do don't hide. You can do so hide line. ठीक है. Hide ribbon. ठीक है. So cartoon. 1 QR this one. So cartoon. You understood this thing?
Yeah.
So we know that this sulfate is interacting with this tyrosine 52 and this argine 263. Okay.
Mm.
Now you can say that this is a substrate binding site. You understood this one? Why I am saying this one is substrate binding site?
Yeah.
Based
Because
on
yeah.
based on this sulfate ion.
Interest.
Based on this
Yeah.
sulfate ion.
Mm.
That is a non-quinone substrate. It is a non-quinone substrate. I am telling you the non-NO and non-quinone substrate. Okay.
Okay.
Now what you do? Superimpose. Just open this one 7VM. Okay.
Mm.
Or you can tell me whether tyrosine 52 is present, because this one is also interest. So just hide first this one keyword. Now tell me tyrosine 50, which residue is interacting, tyrosine 54 or 52? We can do
Tyrosine.
it by technical view, okay, from this one, for this one, okay.
Mm.
Go to action, reset, technical. Now you can see the residue, which residue are interacting. So there is no tyrosine interaction. So there is no tyrosine is not interacting. Where is tyrosine? Display sequence already. Tyrosine 54 is here. This one is not interacting. This one is not interacting. Okay. But which residue are interacting? Let's see. Let's see the result Q which we are getting now.
Yeah, Q48.
Okay. So these are the interacting residue. These are on it.
Mm.
So you can see that this one is interacting with this residue. This one.
Which
This
is
is Q 46, 47, Q 48. Okay.
yeah, Q48.
Storage stick. Storage stick. Okay.
Yeah.
So this is the active this is the active site with
Mm.
respect to this one. There is lot of hydrophobic interaction, so I have not going to look at the hydrophobic interaction. Okay.
So what we gonna write in the table for the conserved residue?
Yeah, I'll tell you, no, wait. So now what you do? Go to this 7VM, hide line. Okay. Hide driven. So pattern. Okay. Just minimize this polar contact of this one. Okay. So we have this active site.
Yeah.
Understood?
Mm.
But this one is interacting with this one. Now you open that one key. Now you can see that here also tyrosine 54. So, so this one is we can't say. If we get the tyrosine
Mm.
interaction, okay. Likewise, sulfate is interaction with tyrosine here.
Mm.
In case of
Sulfate.
KUR, and this one is 1 KUR. Okay.
Mm.
So can't say that this one, if this one is not, and this one is interacting with phenylpheniquinone,
Mm.
but we have not any interaction from this one. If you get the interaction from tyrosine 54 or tyrosine 54 is interacting with this E. coli or tyrosine 54 in my cases. Okay. Now I open my structure. Okay. If you
Mm.
want, I can just hide. I already get the tyrosine 54, huh?
Mm.
So here the sulfate is also interacting in tyrosine 54. This one is interacting, but this is Q40 is interacting here, huh? There
Mm.
is no interaction from that one. What I do now? I just hide this one. Okay. So we have the arginine 263
Mm.
here in tyrosine 52.
Yeah.
Now I will open that unimol B2I structure that I've got, huh?
Mm.
Docked one. 7VM PQ unimol this one. PQ docked. Let's see which one this one. Yeah, this one. Give me a minute, I just check it whether this one has interaction of tyrosine. Yeah, this edge. So I'm just opening this one, okay? The unimol B2 that I got my result tyrosine 54, okay?
Mm.
You can see here,
Yeah.
tyrosine 54 is interacting. Just orientate yourself. So you have to get the interaction conservation likewise. So in that case, your cases, you just orient now your molecule, definitely you'll get one interaction from tyrosine. If you look at the different conformer, okay?
Mm.
Try to get this one, okay? So this one is the phenylpheniquinone. So this one I unimol docked, go to action, reset, technical. Okay, now you can see the tyrosine is interacting here. Where is the tyrosine? So this is the tyrosine is interacting. You understood? One is interacting with the this carbon, this oxygen included this carbon, and this oxygen interacting with this tyrosine. Which tyrosine? Uh, is this one. You can see this oxygen interacting with this tyrosine.
Yeah.
Just sorry, this one. So edge stick. Okay.
Mm.
Now if you superimpose both the chain, na, definitely this tyrosine superimpose with that one also. Okay. Now you go that one. This one is the PQ. You just hide. I just am hiding this one, है ना? Polar contact. This one is so hide ribbon. ठीक है? Hide line so cartoon. ठीक है? You got it?
Yeah.
So I just mark the tyrosine that the active site tyrosine. I just mark in different color so that you can visualize it here, है ना?
Mm.
I just color it through different. Let's say I change it to which color you want. Red or green already yellow already magenta. I'll change it, है ना? Change into magenta. Okay. So this is in case of 7 VM docked PQ crystal docked PQ 7 VM docked Finithian Kiran dock structure, and this is my active site. Okay. With respect to sulfate ion,
So,
with respect to PQ, this is the tyrosine 54. This is the tyrosine. Which residue? This one is the tyrosine 54. Okay.
mm.
51, 52. Okay.
Mm.
So you can rename it tyrosine 54 if you want. You can just rename it label tyrosine residue 54. Okay.
Mm.
I just label it. We'll go to it. Okay, sorry. Just, this is tyrosine 54. You understood?
Yeah.
Now, what you do, because we are looking at the hydrogen bond interaction, not looking at the hydrophobic. You can do the hydrophobic interaction also. Okay. But if you get the hydrogen bond interaction and these are the same in other cases, it means that this one is conserved. Okay.
Mm.
And that is one is very important. Now you just open that one. One QR. Where is one QR? This one. Okay, I have to align that one, na? First, that one. I have to align which one to which one. So this is I have done with the 7 of unimol dot complex. This one is the 1
7
QR.
to Q1Q.
Now I will align to this one QR. Okay, so see one, this one is that one.
Mm.
I will align to this one. Okay,
Yeah.
so I will take this one, align to one QR. Okay,
Yeah.
now you can look at this one.
You can zoom in.
You can zoom. See, no, you can see the zoom. Now you can see both the tyrosine are in the same position and both are in it means that tyrosine 54 is tyrosine 52 in case of which one? E. coli.
E. coli. Has
Or you
no,
can do
no.
one thing. You can do one thing. Hide the carton from both the.
Mm.
Hide carton from here. क्या हो रहा है? How do we move this one? Okay. This one is that this one I opened. Okay. Sorry, this one is that one. Okay. What you do? Hide cartoon from this one. Hide cartoon. ठीक है? And hide cartoon from this one also. Hide cartoon. Now you can see. Just minimize all the hydrogen bond. I don't need this one polar contact hydrogen bond. Okay. This one I don't want. And that one is also polar contact. This one. Okay. This is the alignment actually. I just look at the alignment. Now see the NAD page here. Okay, sorry. Just you remote, click it here and then rotate it, zoom it or orient it. Okay. I can see the NADPH of both the cases are aligned
Yeah.
little bit. There is deviation. So this is the difference, real difference that in that case. But see that both are very much little bit deviation, है ना? So this one is crystallized with some different conformation in NADPH, and see the adenine ring are slightly shifted. But we are not interested in that one because that one is experimental structure. Okay,
Mm.
but we know that in case of E. coli. Okay, in case of E. coli, we know that this sulfate, this is a sulfate
Mm.
of this one. This is a sulfate of this one QR, and this PQ is of 7 VM, this one. Okay. So we know that the sulfate, what are the sulfate interacting site? Till
Tyrosine 52, arginine
this
263.
sulfate. So this is the tyrosine. Sulfate is interacting with this tyrosine and this arginine. Okay.
Yeah.
Form one QR structure.
Mm.
Okay. Now we know that phenethylene quinone is also interacting with this nicotinamide of that one, 7 VM.
Yeah.
But phenethylene quinone is also interacting with this tyrosine.
Mm.
But having in some different orient, little bit different. But this one is interacting with this one is interacting with this sulfate.
Mm.
Okay.
Yeah.
It means that both are the same substrate binding site. You
Mm.
understood? You can get it my concept or not?
Yeah.
So in this way, tyrosine 54 is conserved.
Mm.
So you can even look at here alignment. Okay, both the alignment in the same part, same residues. So see, we'll select this one. Okay, go to editing mode. You just select it here. You just select it here and see both are following in the same path, up
Yeah.
and down. See, both
Mm.
are. It means that. So I already told you with respect to sulfate, we
Mm.
predict this is the for E. coli. But
Mm.
we have docked to verify we don't because this is not a non-quinone this is not a quinone substrate now.
Mm.
So if you slide one QR structure, definitely one QR quinone will sit here. You understood?
Yeah.
And if you slide PQ with 7V, if you slide with phenethylene quinone definitely sit here.
Yeah.
So this one is the active site.
Mm.
This one is the tyrosine 54. It is a putative active site.
Mm.
You understood?
Yeah.
So make a table. So you do it in the same way for other one also.
Mm.
You understood? If
Yeah.
you open the other picture now, you can get the same type of. But there is some problem with the HJ6C. We don't know. No, you have to dock NAD page and then PQ. But
Mm.
you open other one tyrosine also come here also. Let's open other structure. All all having, but we don't know whether that structure having some molecule or crystallized here present or not. Okay,
Yeah.
then we can't say because this one is interacting with not that one
Mm.
sulfate and that one is also interacting. In this way, you can say this is so some other structure if they crystallize with some this molecule, other molecule, they are also interacting with here. So we can say this is a putative substrate binding site in all the cases that are conserved. You understood?
Yeah.
But you have to do it for the other hydrophobic interaction. Also, you have to do it for the other one also. Look at the conservation. Like if say this one is interacting hydrophobic interaction and this one is interacting hydrophobic and this one is also interacting hydrophobic. If they both are the same. So it means that hydrophobic one is the conserved one. You understood?
Okay. Out of
Not
the,
done.
yeah.
You can do it simple. You can just select it and
Mm.
just make a command line so stick by rest by 4 Armstrong or sulfate. So stick by rest 4 Armstrong or PQ. Understood?
Okay. Well, that's for,
So in this way, you have to write in the table. This is the putative substrate binding site. Tyrosine 54 is conserved.
mm.
Here tyrosine 52. That one is conserved. If other one is like hydrophobic, some are hydrophobic, all are matching. So that one is conserved. Some are not matching. That one not conserved. So make a table one as likewise. So you can make a table one PDB, second PD, third, fourth, fifth. What are the hydrogen bond interacting residue? What are the hydrophobic residue? If all the hydrogen bond interacting residue, okay, are same,
Mm.
that is conserved. If not same, they are not conserved. Understood?
Okay. Yeah.
So if you want to open other structure, I can show you the other one also in the same window. Go and open other one. Like tell me some 4R view. Let's see 4R view because 4R view also is a sulfate ion. Okay. 4R view. Okay.
Mm.
Oh, okay. It's not link fetching. Okay. This one is 4R view.
Mm.
So you have to always take one QR as a reference because this is the experimental one. Okay.
Mm.
Now I just remove everything from 4R view. take one chain only. Go to chain. Okay, sorry, this is view mode. Go to chain. Remove everything. Remove the B chain, A chain, C chain we don't want. Where is the C chain? This is D chain. This is B chain. We don't want B chain. We don't want C chain. We don't want D chain. Okay. Remove water molecule. So we want only take the electron accept. Okay, then we can say this is a substrate, but this is a non-covalent. But we can look at this one. We take this one as a reference molecule and look at the conservation. Okay, so definitely we'll get the tyrosine here. Let's see. So I will
Remove the
remove the
chain.
chain. Just go to X and remove water molecule. Okay. Now you remove. We take the one and I will just keep this one. Huh? This one is P or which one? GLO. This one. Okay, look at this one first. Look at the. This is a tetramer. Huh?
Mm.
Now you can see the tetramer here. Okay. So which one is A chain? Okay, look, color by chain. I will do it color by chain. Okay. We have changed the color. Now I will remove everything. So this one is this one I will keep. No? So everything is selected. Here also A is selected somewhere else. Okay. So we want this one and look at this one. Where is glycerol? Zoom it and see this one. I don't want. Okay. So I don't want to delete this one. One having glycerol, one having EDO actually. This
Oh,
is ethylene glycol. This one is different. This is having different one. One is crystallized with this one. Okay. And someone is different. Someone was different molecule actually. So, okay. All are with this molecule. Okay. All are with this molecule.
so which one is better, glycerol or EDO?
All are glycerol. No? This is glycerol. This is glycerol or glycerol. There is no EDO here. EDO and it can 7 be important. Okay.
Oh, yeah.
So this is you can rename by chain if you want. Label by chain. So this one is A chain.
Mhm.
So I will remove all the thing from this way. I don't want to remove these three. This one I don't want to remove. Okay, just select it. Just remove it. Okay, just select this one and this one. So I have removed all the thing. Okay,
Mhm.
now this one I don't want this from this one is remaining. Okay, now hide the label. Now I will superimpose. This one I also don't want. Okay, there is some other molecule. I just need this one. There is some other molecule, just select it, remove it. Okay, now I can look at the interaction here. What are the interacting residue? Go to this one is already select done. Okay, go to action, preset, technical. See if they are interacting with or not. What residue they are interacting. So you can see that this one is interacting or not. Yeah, okay. Tyrosine is interacting here also. See, consume it, select it, and orient it. Okay, now you can see here.
The difference between technical and choosing the polar to suit the polar contact.
Yeah, I can do it in that way also. So this one is, see this one, tyrosine is here. One is interacting with this residue, okay, and this one is interacting with two residue here. They are interacting. One is tyrosine C, this one,
Mm.
okay. Three residue actually they are interacting. You see, this
See.
is interacting.
Yeah.
Save it, okay. Residue, select. You can select this one,
Mm.
okay, and then you can select this residue also is interacting this one,
Mm.
okay, and then which one? See, this one is also interacting this residue. You
Yeah.
can see this one, this residue, this one, this residue one. These
Mm.
are the three reactions with that one, another one or nothing. Okay,
Yeah.
now so it stick.
I have different kind of bimol. You have
Means
bimol 0 and
okay, there's something they are different type of palmol 1.1, 0.1. Now I've selected this three residue. Hide the cartoon, hide cartoon, hide line, hide ribbon. Okay, polar contact. Okay, now as you can know these are the interaction with ADO. Okay,
yeah.
just label it if you want. Label residue. Now you can see tyrosine is it means the tyrosine is conserved in all the cases. You understood?
Yeah.
Now you can see arginine is here. So arginine is conserved in two cases, I think. Yes, it tyrosine. Okay, now just superimpose to one QR because you are taking one QR as a difference. Okay,
Yeah.
you can superimpose to one each other if you want. If you superimpose this one to one QR. Okay,
Mm.
now we have this one. Now just want to superimpose align to one QR.
Mm.
Now where the structure go? See this one is from here. Okay, you want you can hide this one. This polar, this polar, this polar, and this polar contact to dot net. Where is the other polar contact? This one, polar contact. This polar I already hide. This already hide. Where is the polar other? Okay, forget about polar contact. Now you can see here. Where is the tyrosine? See, tyrosine is conserved
Mm.
and arginine is also conserved in two
Yeah.
cases. And see, you can hide the NADPH if you want because we know na already this thing na.
Mm.
So see the NADPH are overlap to each other all in all the cases.
Yeah.
Okay. Now two cases with respect to if you okay, I will do one thing. Just hide 7 VM here.
Mm.
7 VM. This one I hide. Okay.
Yeah.
Okay. You
Okay.
can see the EDO and equilysol bind very much close to each other. And the tyrosine is conserved in both the cases, in two cases.
Mhm.
So you take the, I have taken one QR as a reference. What you do, you take one YB5 as a reference.
Mm.
Okay. So in
Human.
this way, you can understood that if you take one QR as a reference, and if you look at, if you do the conservation
Mm.
with say 3QBA, then we can say that, okay, so this one is the, we can say these are the substrate binding site.
Yeah.
Okay. We, but we don't mention about this molecule. We only say these are the putative substrate binding site. If
Mm.
someone asks, then we can say, okay.
Okay.
So you can say that even you can look at the alignment, see the tyrosine 52 and tyrosine 15 both are come here.
Mm.
That is the alignment. So we have to do the alignment. There is something on Ella 3Q2B2, this one. If you
Yeah.
click this one,
Mm.
okay, where is why this not happening? 3Q polar. 3QBA. This is the alignment. Okay. Now, okay, why did it not align?
You have to write a line again.
Because this is glycerol come here and lipase come here. E come here. That's why making this doing. But you can understood now.
Mm.
Definitely, if you export the alignment, now you'll get this type of alignment. Okay. Okay. Why that happening? We have to show is complete step. We have not shown this one.
Yeah, why?
And maybe show it cartoon. Okay. And which one? One QR. This one. So it's cartoon. Okay, I've done this one. Now just do the command once again. Let's see where is the why that is happening. Now I'll tell you. So this having very less identity. That's why see this one is NADPH here, glycerol here. That's why it's not aligning in this way. And sulfate here. Okay, sulfate from which one? One QR. Okay, sulfate here. Sulfate NADPH this one for HN and we have glycerol and so because this is FA written, na, that's why. So don't get confused. Na? If you do the alignment, na, if you take this, if you export the alignment, wait, I will tell you one. You have marked this one, Arginine 263 and Arginine 270. Okay,
Arginine
these two are conserved
263 and arginine
270.
270.
So these two are in the same position. If
Mm.
you click, do one thing, you just have to polar contact this one as well. Okay, this is one is the already aligned. You just click it. Which one is clicked here? This one. Okay, this one already clicked. Now you can see here same residue or not. See arginine 2 now you can hide the carton
How
if you
did you
want.
align again?
Or there is something here only. If you click here, na some aligned. That's why that one in this one is alignment.
Mm.
This one is alignment.
Mm.
Okay.
Mm.
LN 3QW 1Q और हम just click करे थे ये वाला.
Mm.
Okay, that's why this one is alignment.
Yeah.
Okay, just you have to click it to get the alignment. Now you just hide the cartoon.
Yeah.
Cartoon from here, hide cartoon. Okay, and then you hide cartoon from here. 1KR. Okay,
Yeah.
now you can see the same thing in the sequence alignment file. You also get it.
Mm.
Now you can see here the tyrosine 52 is conserved with respect to this one and this one. Okay.
Mm.
With respect to arginine, this one and this two are conserved also in both the cases, except this one is not conserved and this
Yeah.
one is not interacting. Okay.
Can
This one is
someone
interacting to but one, this one is interacting to someone, but there is no corresponding data. You can't say this one is conserved. Okay.
Mm.
There is no corresponding interacting, but these two are interacting. This one is interacting this one. Okay.
Yeah.
That's why you're taking saying that if you look at the conservation, look at the interaction also, which one is interacting.
Mm.
Now you can look at here. If you click here and here, sorry, editing mode, if you click it here and here, now you can look at here. See the arginine, two
Yeah.
fall in the, these are conserved. Now you can see here, tyrosine, this one and this one.
Mm.
See, this one is conserved. Understood?
Yeah.
Now you open the third one to this one, 7 BM, PQ docked. Got it?
Yeah.
Now you understood from these three
Tyrosine is conserved.
in
And
all the cases.
yeah.
Okay. So here there is no arginine.
Mm.
Here only this tyrosine is interacting with this one. Okay.
Mm.
But here tyrosine is conserved. But you can't look at the alignment file three at a time here. You have to only look at the two at a time. Either you have to hide this one or hide this one. Okay.
Yeah.
Because pymol only give you the peer alignment.
Mm.
Okay. Now
Mm.
you can do one more thing. Take the uniprot sequence
Mm.
for all of the sequence protein and do the cluster omega. Then in that file, you'll get the tyrosine 54 in the same position. Okay. So
Yeah.
see, this is tyrosine 54 here, tyrosine 52 here, and tyrosine 59 here. If we do the sequence alignment, in the sequence alignment, in the third three sequence, in tyrosine is falling or in all the same cases. Likewise here, consider this is tyrosine, tyrosine, and tyrosine. In the
Mm,
sequence, if you do the cluster omega and then then these three come in the same place. Understood?
mm,
In the sequence alignment.
yeah.
So in this way, you can find the conservation.
Mm,
You understood or not?
yeah, I
One
got
with respect to sulfate, one we have already docked.
it. Mm,
Okay. If we don't dock, let's say we don't know this one, then how do you know? We can take the sulfate as a reference molecule.
difference, yeah.
And because in sulfate there is a possibility to transfer the electron. We don't know whether EDO has a possibility to transfer the electron or not. Okay. So we verify our result with the help of this 7 VM. In 7 VM, we will get this one. And if we don't know this one 7 VM, that what we will do? We will take the sulfate as a corresponding residue. And what are sulfate corresponding residue? Here also you can get the sulfate corresponding residue. Tyrosine 54, you will get it.
Mm,
Okay.
okay.
So it's based on, we can say that this one is conserved, but we don't know which one is interacting. But we can say based on sulfate ion, these are the interacting tyrosine 54. Now we have verified by docking the quinone. We get the tyrosine here. You
Yeah.
understood?
Yeah, understood.
Now so you have to do this way, and you have to write the conservation for all the homologues.
Mm. Click the
You understood?
click the picture as well and do.
I will share the screen for a session file if you want.
Okay.
Now you understood now?
Yeah.
What you do? Forget about everything. Okay.
Mm.
Take only one QR as a reference molecule.
No,
Because
one
if we
AB5 as is.
one QR as a reference molecule. Or what about one YB5? One YB5 is crystallized with a sulfate ion. Let's see.
Mm,
H one YB5. One
B5.
YB5. Okay. Just enter it. Okay.
Mm,
Now do the same thing for this one. See, this one is also crystallized with sulfate ion.
yeah.
Okay. It means that we have a reference here also.
Mm,
You can compare. Let's see the conservation with respect to this one.
mm,
Okay.
yeah.
And let's see whether in case of 7 we get the tyrosine, we can see whether we can get here the tyrosine 54 or not. When the tyrosine is very, very important residue on Rust. If we mutate the tyrosine 54, maybe activity will loss.
Yeah.
Now let's work for this one. Go to action, remove water. Okay. Now this
How
one
to,
is sulfate bond. Okay.
yeah. Remove chain.
Here we have N. Okay, this one is A. We just remove to B chain. B will remove everything.
Mm,
Just remove everything. Okay.
mm,
Now we have get this one.
yeah.
We don't want this zinc ion or something, some other acid we don't want.
Mm,
We know that, and it is written in the paper that it is possible that the electron get transferred from here to here.
mm,
Okay.
okay.
So if the electron get transferred from here to here, this means that this act as a substrate, because
Mm,
if something there is transfer of electron to the, likewise it's transfer of electron from quinone, na? So
mm,
quinone get converted into hydroquinone. Now here the electron there is a transfer of hydride ion and get transferred to here. In this way sulfate get converted into some other molecule.
mm.
In this way sulfate act as a non-quinone substrate. You understood this point, na?
Yeah.
Now because it is written clearly in the paper that there is a possibility that here to here the electron, but we don't know about EDO, whether the electron, there is nothing written EDO in case of electron get transfer or not. You read the EQR paper, it is written, है ना?
Mm,
One QR paper. This thing is written also in for our VU also. We reference taken the reference one QR. Okay.
mm,
Now you can look at the interaction here. Same thing do. So we are looking at the putative substrate binding site. of this protein, because with this protein not crystallized with benzoquinone, naphthoquinone, phenylquinone. Okay, or alkynyl, they get the activity with alkyl, है ना? So
Mm.
if you dock the alkynyl, you get here
Yeah.
alkyl come here, है ना? Now you can do the same thing. Look at the interaction here, but we have the reference.
Hello.
This meeting is being recorded. Okay, now you can hide cartoon, hide lines, hide ribbon. ठीक है, polar contact. Okay.
Yeah.
Now this one is not required. This one is from some other model. Now you just align to one KUR. Align into, or you can align to this, because this one is also a reference. You can align into, let's say 7 VM, or you can align into 1 KUR. Okay, because this is already a reference one, sulfate ion is already present. Let's align this one, or you can align to 7 VM. Which one you want to align it? Let's align to 1 KUR. So we have aligned everything. Okay, ठीक है.
Yeah.
Now you can see tyrosine is conjugate. You understood or not?
Mm.
What you understood now from this way, you can write a PhD project. Just remove this one. ठीक है?
Mm.
Convolute. Now you can see here. Everything is understood. That arginine in these two cases are conjugate, and there is nothing interacting na here. So, but tyrosine is conjugate in all the cases. So if you get the docking with 8J6C and you get the tyrosine also,
Yeah.
it means that docking is correct. If
Mm.
you get the HSC dock with NADPH and dock with phenanthrene quinone, naphthoquinone, benzoquinone, try docking with the phenanthrene quinone always. Okay. And if you get the tyrosine 54 in 8J6C also, hypothetical,
Mm.
it means that, because in case of that one, 7 VM, we already confirmed it by paper that tyrosine 54 tyrosine is interacting. Okay.
Mm.
Uh, I already, so we also confirmed by docking also, huh, whether they
Yeah.
have written correct or not. Okay,
Mm.
because they get the activity with that one. Okay, so this, see this one, tyrosine conjugate. Tyrosine conjugate. You have to open. So like you have to written in the paper. You just removed all this thing. Sulfate, you have to just remove it. This phenanthrene cannot just dock, remove it and make this one, this figure only. This figure, this one. So we can make in paper that this is conjugate. This one is conjugate in two cases. One is not. Okay. Just where there is no corresponding residue, just blank it. That one is not conjugate, or you can look at the what are the other residue that are corresponding to that one. That one is different one. Okay, you can write it if you want. Oh, no? Because
Mm.
you have to write the same table, same residue, no? Same position in the residue. Okay, aligned residue.
Yeah.
Likewise, you get the tyrosine 54. You write it tyrosine 54, tyrosine 50. What are the this one tyrosine 59? Okay, tyrosine 59, tyrosine 54, and this one is 59, 52. There are four tyrosine. No? One is which one is tyrosine 52? One is which one? This one, not selected.
Arginine.
Label residue. Okay. So there are three tyrosine. Just one I have not labeled it. Label residue, one letter residue. So see, this one is not labeled. Tyrosine 59. So tyrosine 59, tyrosine 52, tyrosine 59, tyrosine 54. It is in the same position. Okay.
Yeah.
It is in the same position. You select this one. Okay. This one and that one. One is not selected. This one. Okay. So these four are in the same alignment. If you do the alignment, you do the one keyword that 7 VM, this come here. Okay. If you do this one to this one, this come here. Okay.
Okay.
If you do with this will come here. But if you take the sequence, okay, of all the sequence, okay,
Okay.
and do the sequence alignment, then you will get that this tyrosine 54, this
Line.
tyrosine, these all are in the same
Line.
line. You understood this thing, now?
Yeah.
Now, which one is remaining? I have done with 1 KOR, 7 VM, 4 RBU, 3 QBU. Which one is remaining?
3 TQH. 3 no. TQH.
Yeah, you can try. I will share this session file. Na? You can just open all that and let me
Yeah.
know which one. Okay,
Okay.
I will share this session file. Okay, let meet tomorrow. Okay.
Okay.
Now you understood this thing that this tyrosine is very important.
Yeah, it's conserved, and if mutation occurs in this residue, then the
The protein lose the activity.
function, it loses function.
Either the function will either the activity
Structure,
will
yeah.
either the activity will go down or maybe the function will completely abolished.
Mm.
Because this is so because we know that no one have crystallized the protein structure है ना, with the ligand. If anyone have crystallized it, then definitely this one is interacting.
Yeah.
We already verified it from this docked structure. We got this one with respect to this one sulfate ion. We got this one है ना.
Yeah.
So this one is a substrate. This is a conserved putative substrate. Why we say the term putative? Because no one have crystallized the protein with substrate. You understood?
Yeah. Got
Like
it.
we have dog this one paper at 7 BM author also have dog this one. They have not get the crystal. Here they have get the sulfate but not crystallized with the phenanthrene benzoquinone diphthocquinone. No?
Yeah.
Likewise 1YB5, if you look at 1YB5, they have taken get the activity with the alkenyl. So if you take the alkenyl and dock it, so here the alkenyl also dock here only. Try that alkenyl one. Oh, okay. Read that one way before they have taken what PubChem ID and take that one and dock it. You can also get alkenyl dock here. है ना?
Okay.
And if you look at the interaction, definitely tyrosine will also come. Alkenyl bind here and they interact with tyrosine 54. They have written in the paper tyrosine 54 in there they have written also. I can show you if you want. Can you share me the PDF 1 by B5 paper?
Yeah. You have said.
I have sent in the WhatsApp now.
Yeah. Kinetic wala. Point
Yeah,
one, yeah.
yeah. Can you send it again?
Okay.
WhatsApp. Can you send it again? I will to open the PDF and show you. I
Okay.
have to find the paper.
But
You just
I haven't have food. I think we should continue.
I want to show you that one tyrosine 50 pound is there mentioned on.
Mm.
Wait, I just finding the paper. Have you find the paper 1 by B5? That kind of paper where it is? Can you send me again?
2 minutes. Yeah.
Okay, so one is forward it now in WhatsApp now.
Done.
Done. Na? You have you seen? Where is? Okay, yeah. Wait a minute, I will show you. I have to share my screen. Can you see now paper?
Yeah.
So they get the first in the human Zeta-Catlin, they have get the activity with some benzocaine naphthalene. Later they have did the activities this one also.
Mm.
Okay.
Mm.
So they propose the mechanism based on this one.
Mm.
So if you read the activity, na, they have definitely mentioned that they get the activity with the protein. See the kinetics. So they have get the activity with, quinone. Quinone, they also get the activity from quinone also.
Mm.
Quinone, 2 alkenyl, they get the activity and 3 alkenyl. You just take this 3 one, 2
Mm.
alkenyl and to this one you can dock. है ना?
Yeah.
I will show the result here. They have docked the structure.
Mm.
Now you can see this one. 2 alkenyl,
Mm.
2 hydro, 2 hexenal and 3 nonol they have taken and
Mm.
dock it here. So this is the 3 nano en. See tyrosine come here. Okay.
Yeah, yeah.
See this one, this molecule, this one.
Also
See tyrosine
tyrosine. Yeah.
59 come here
Yeah,
and see
again
this one.
tyrosine. Yeah.
In that the tyrosine is a putative conjunctive. Here it is, they have also dock it now. So
Mm.
this one is putative substrate binding site
Okay.
based on this means that we tyrosine 59 or that if you take 8G6C and mutate tyrosine with respect to that one, you get
Mm.
the, even though you have not crystallized the protein with the sulfate NADPH uh, and substrate molecule. But based on this study, if your 8G6C get the active, if you get the activity, है ना?
Mm.
Okay, by chance you have get the activity with L Donovani. Now if you mutate Y59, based on this study, your protein activity definitely abolished. Understood?
Yeah, got it.
So see, this one is tyrosine 59.
Yeah.
So you can mention this tyrosine 59 is conserved. Okay.
Okay. Okay.
And here I can also you can here also you can see that the primal that one is also
Conjure.
59 conserved. You understood now?
Yeah, understood.
Like that you can do it for the remaining one.
Mm.
Okay. Now you make a table
Yeah.
with respect to substrate binding site and with respect to NADPH binding.
NADPH funding side.
Okay.
Okay.
Got it. I will share this
Okay,
session
got
file.
it. Yeah.
You can do it here also, here in this same session file. Okay.
Mm.
Your concept now you are getting the substrate conservation with respect to substrate binding site.
Yeah.
Now you can make the table. Okay.
Okay.
You have to write all the information in your review paper.
Mm.
Okay. Bye bye. Good
Bye
night.
bye. Good night.
I'm sharing the session file. Okay.
Yeah, sure. Bye
I
bye. I'm ending the meeting.
okay. I'm sharing the session. Where you just close the meeting and share it.